Hello, habari zenyu? How are you? Welcome back. So, in our continuous series of the carbohydrate metabolism, let us now look at the tricarboxylic cycle. If you've remembered, we have been looked at glycolysis, we have looked at the pyruvate, and we said one of the fates of pyruvate is that it was converted to acetyl-CoA in the TCA cycle. And we mentioned then the acetyl-CoA is continuously broken down in the TCA cycle. If we look at the 6 carbon of glucose we had, we have already lost 2 in the pyruvate dehydrogenase reaction. And so now we have 4 carbons remaining from the glucose molecule in the form of 2 molecules of acetyl-CoA. Let us look at what happens to them in the TCA cycle. The TCA cycle is a very interesting pathway, also known as the Krebs cycle or the tricarboxylic acid cycle, as it is the final pathway where oxidative not only of carbohydrates but also of amino acids and fatty acids converge. In the cycle, the carbon skeletons are converted to carbon dioxide and the reducing equivalents are transferred to oxygen to make water. So this particular part, the TCA cycle, provides most of the energy from the catabolism of carbohydrates. And yes, it occurs in the mitochondria, very close to the electron transport chain, so that the reducing equivalents that are carried by the coenzyme FAD and NAD are taken to the electron transport chain. And it is also an aerobic pathway, occurs exclusively in the presence of oxygen. So we can think about of the reactions of the TCA cycle in 2 phases, the first 4 and the last 4. The first 4 reactions, generally, they cause the loss of the 2 carbons of acetyl-CoA in the form of carbon dioxide. Let's look at this first 4 reaction. So we have acetyl-CoA that has come from the pyruvate dehydrogenase reaction or from other sources. We are going to see under amino acid or lipid metabolism or fatty acid or metabolism. And this acetyl-CoA in the first reaction condenses with an intermediate of the cycle called oxaloacetate. So in the first reaction, we have a condensation reaction where a 4-carbon oxaloacetate condenses with the 2 carbons of acetyl-CoA to give you a 6-carbon molecule known as citrate. This reaction is catalyzed by an enzyme called citrate synthase. Even though it's a condensation reaction and we have creation of a new bond, we do not require energy because the splitting of the bond between the carbon and sulfur of coenzyme A provides enough energy to allow the condensation reaction to occur. So the first reaction is a condensation reaction and we have released coenzyme A. So the next reaction, catalyzed by an enzyme called aconitase, is what we refer to as an isomerization reaction, where the molecules of citrate rearrange themselves to form what we know as isocitrate. If we can look at what is rearranged, look at this hydroxyl group. Initially, it was on carbon number 3, but it is now on carbon number 4. So this is what we refer to as isocitrate, and it is catalyzed by the aconitase enzyme, and it is an isomerization reaction. In the third reaction is what we refer to as an oxidative decarboxylation, and at this point we start losing the carbon dioxide that were brought by acetyl-CoA. This oxidative decarboxylation is catalyzed by isocitrate dehydrogenase, and it is called oxidative because we lose electrons which are carried by NAD. The oxidized form of NAD accepts 2 electrons to become NADH plus H. In the process, our 6-carbon molecule of isocitrate loses 1 carbon as carbon dioxide to become our alpha ketoglutarate. Okay, so here we have lost the first carbon dioxide. The next reaction, the fourth reaction, is also referred to as an oxidative decarboxylation. Again, just like similarly we saw, we lose a carbon dioxide, the second carbon dioxide. Okay, however, This enzyme, alpha-ketoglutarate dehydrogenase complex, is named similarly to the pyruvate dehydrogenase complex because it has a very similar structure. One is that it's made up of 3 enzyme subunits like we saw in the pyruvate dehydrogenase complex, and it also requires the 5 coenzyme TPP, NAD, FAD, lipoic acid, and coenzyme A. Similarly, the end product of alpha-ketoglutarate is attached to coenzyme A, and this time instead of acetyl-CoA, we have succinyl-CoA. And we also lose a carbon, and so our 5-carbon compound of alpha-ketoglutarate remains as our 4-carbon compound of succinate, which is attached to coenzyme A, and refer to it as succinyl-CoA. So you can see the major thing that happens in the first 4 reactions. We have 2 decarboxylations, and we lose the 2 carbons of acetyl-CoA, and we have generated 2 molecules of NAD, NADH, and formed a 4-carbon compound of succinyl-CoA. The next 4 reactions, we can think about them as replenishing or going back to oxaloacetate, which was the initial step where we started. So the first reaction is what we refer to as the substrate level phosphorylation. We have succinyl-CoA attached to CoA, and we saw the carbon-sulfur bond. When we split this bond, we generate enough energy here to condense 2 molecules together. In this case, the energy that will be obtained from splitting succinyl-CoA from splitting the CoA compound from succinyl to release it as CoA will be enough to condense GDP and an inorganic phosphate to give us GTP. And this GTP immediately gives this phosphate group to ADP to form ATP. So here is what we refer to as a substrate level phosphorylation, because in this step we synthesize 1 molecule of ATP, okay, in the form of GTP, which is equivalent to 1 ATP. This enzyme here is called succinyl-CoA synthetase. So now our end product will be succinate. The next reaction, number 6, is oxidation of succinate, which is catalyzed by the enzyme succinate dehydrogenase. If you remember the electron transport chain, this was complex 2 of the electron transport chain. And so succinate loses 2 hydrogens. It is reduced, or it's oxidized, sorry. It is oxidized, and these hydrogens together with their electrons, they're accepted by FAD to form FAD2. And now oxidation of succinate yields fumarate. The 7th reaction of the TCA cycle is a hydration reaction where water is added to fumarate to give us malate. If we look at carbon number 2 here, the water molecule joins here. Remember here we have a double bond between carbon number 2 and 3. And so the water molecule joins carbon number 2 to hydrate it to form an alcohol group, okay, an OH group. And so we have malate, okay. And this enzyme is called fumarase, which catalyzes the hydration. The last step that generates oxaloacetate is another oxidation where we have the hydrogen groups from the second carbon, and the electrons are accepted by NAD to generate NADH, and we have back oxaloacetate. Oxaloacetate who will now be ready to combine again with acetyl-CoA to combine with the cycle. So we can see in the second phase of the cycle, not only have we generated a molecule of ATP, but we have generated 1 FAD and 1 NAD. So in total, a full cycle of the TCA cycle, it generates 3 NADH, 1 from the isositol dehydrogenase reaction, 2 from the alpha-ketoglutarate reaction, and 3 from the malate dehydrogenase reaction. It also generates 1 ATP from the succinyl-CoA synthetase reaction and 1 FADH2 from the succinate dehydrogenase reaction. In the process, the carbon dioxides that were in the acetyl-CoA are lost. So how much energy do we get from the TCA cycle? Remember we mentioned that the TCA cycle occurs in the mitochondrial matrix. in close proximity to the TCA, to the electron transport chain. And so all the reducing equivalents carried by NAD and FAD are transferred to the electron transport chain. And from the lecture of the electron transport chain, we saw that each NAD, when all the electrons from NAD are transferred to oxygen, we generate 2.5 ATPs. When all the electrons are transferred from FADH2 to oxygen, we generate 1.5 ATPs. So in that in mind, we know the three NADs from the TCA cycle will generate 7.5 ATPs, while one FADH2 will generate 1.5 ATPs. And remember, we had one GTP generated from the succinyl-CoA synthetase reaction, okay, succinate synthetase reaction, and we have one ATP. So if we add all this, we know a total turn of the TCA cycle will generate 10 ATPs. However, if you remember this lecture in terms of glycolysis, we know the glucose molecule was split into 2 molecules of pyruvate, and these 2 molecules of pyruvate were converted to 2 molecules of acetyl-CoA. So in this phase, the TCA cycle for glycolysis, we are going to yield 20 ATPs because each acetyl-CoA will enter one cycle, 10 ATPs, another cycle, 10 ATPs, which gives us a total of 20 ATPs if all the acetyl-CoA from glycolysis gets into the TCA cycle. So how is the TCA cycle regulated? Remember, the purpose of the TCA cycle is to provide the cell with energy in presence of oxygen. So there are two phases of regulation where we have key enzymes of the TCA cycle regulated. One of the key enzymes is the citrate synthase enzyme, which was the first enzyme that condenses oxaloacetate with acetyl-CoA. So the citrate synthase is inhibited by high energy levels, as indicated by ATP and NADH. It also receives feedback inhibition by its end product citrate. So if you have a high concentration of citrate, which we will talk about as a high indicator of high energy levels in the cell, it will do negative feedback on citrate synthase. The next enzyme that is regulated in the TCA cycle is the isocitrate dehydrogenase enzyme, which converts isocitrate to alpha-ketoglutarate. Again, this enzyme will be stimulated by low energy levels as indicated by higher levels of ADP and higher levels of calcium, especially in skeletal muscle, while it is inhibited by high energy levels as indicated by high levels of ATP and NADH. Alpha-ketoglutarate is the third enzyme that is regulated in the TCA cycle, and it's activated by calcium, especially in skeletal muscle. We mentioned that high levels of calcium indicate increased requirement of energy in skeletal muscle because calcium is very important for contractility of skeletal muscle. Alpha-ketoglutarate is also inhibited by high energy levels as indicated by NADH and ATP, and it also receives feedback inhibition for its end product, succinyl-CoA. Other factors that influence the TCA cycle is generally the energy status of the cell. As we have seen, high energy status as indicated by high ATP levels and NADH levels are going to inhibit the TCA cycle, while low energy levels as indicated by ADP or AMP or requirement for a lot of energy as indicated by high calcium levels in skeletal muscle will stimulate a higher demand of energy or will signal a high demand of energy and will activate the TCA cycle. So this, if you remember, the main purpose of the TCA cycle is to provide energy. So you'll understand when it is activated, when we have low energy levels or we have increased demand for energy, and it's activated by it's inhibited by high energy levels. Besides providing, we think about the TCA as having an amphibolic nature. So what does this mean? The amphibolic nature of the TCA, because it's a central common pathway, is that it has both catabolic and anabolic functions. Its catabolic function is mainly in the production of energy, where we have seen TCA cycle is able to oxidize acetyl-CoA carbon dioxide, and in the process generates ATP, NAD, and FAD, which all are used in the electron transport chain to provide us with energy. So this is the main role, one of the major roles in catabolic functions. But in addition to its catabolic role, The TCA cycle also has an anabolic role, where its intermediates, they serve as precursors for biosynthetic processes. Citrate, the first intermediate that is formed from condensation of acetyl-CoA and oxaloacetate, is used as a source for fatty acid. It provides a precursor for fatty acid biosynthesis. We are going to see this. Alpha-ketoglutarate can be used for synthesizing the non-essential amino acid glutamate, which can also be used to synthesize another amino acid, glutamine. It also serves as a precursor for the neurotransmitter GABA, which we know is an inhibitory neurotransmitter. Succinyl-CoA can serve as a precursor, is one of the requirements for heme synthesis. And we know heme, we find it not only in hemoglobin, but also in the cytochromes, like the ones we saw in the electron transport chain and other heme proteins. Oxaloacetate serves as a precursor for the amino acid aspartate. Okay, once oxaloacetate is aminated, it will form aspartate, but also it serves as an important precursor for gluconeogenesis, which I have mentioned is synthesis of glucose from non-carbohydrate precursors. Since the reactions of the TCA cycle can be used to synthesize other intermediates, they can easily be degraded. So we have anaplerotic reactions which fill up or replenish the TCA cycle intermediates. Okay, so we've seen things like oxaloacetate, citrate alpha-ketoglutarate, and succinyl-CoA can be used for biosynthetic reactions. How does the cell ensure that these intermediates are not depleted so that the cycle is present? So we have anaplerotic reactions. Some anaplerotic reactions are like the pyruvate carboxylase reaction, in which pyruvate is carboxylated to give us oxaloacetate. Okay, and this pyruvate might come from catabolism of amino acids. It is carboxylated by the pyruvate carboxylase reaction and gives us oxaloacetate. Deamination of aspartate will give us also oxaloacetate. We have another reaction, PEP, or phosphoenol pyruvate carboxykinase, which also generates oxaloacetate. This is one of the reactions of the gluconeogenic pathway we're going to look at, and it also replenishes oxaloacetate, which can now be used for gluconeogenesis. Catabolism of odd-chain fatty acids and branch-chain fatty acids, both of them, their catabolism yields propionyl-CoA, and propionyl-CoA, its catabolism gives us succinyl-CoA, which replenishes succinyl-CoA, which has been used in anabolic reactions of heme biosynthesis. And then we have the glutamate dehydrogenase reaction. The glutamate dehydrogenase reactions generates alpha-ketoglutarate from glutamate. So the glutamate dehydrogenase deaminates glutamate to yield alpha-ketoglutamate. And as you can see here, glutamate can be derived from amino acids. We're going to talk about more these reactions, glutamate dehydrogenase, the pyruvate carboxylase and PEP carboxylase reactions, and even metabolism of odd-chain fatty acids in the forthcoming lectures as we discuss amino acid metabolism, gluconeogenesis, and lipid metabolism. There are some compounds that are able to inhibit the TCA cycle, and they do this by inhibiting the enzymes of the TCA cycle. One of them is arsenite or arsenic, which binds lipoic acid, which we have seen is an important cofactor for the pyruvate dehydrogenase reaction and the alpha-ketoglutarate reaction. So inhibiting this enzyme will in turn inhibit production of ATP. A metabolite of arsenate behaves like inorganic phosphate. So in reactions where we require inorganic phosphate, we have arsenate inhibiting and it's being replaced there. And so we'll find it can inhibit the succinyl-CoA synthetase step by binding to GDP instead of inorganic phosphate. And this will inhibit formation of GTP. And so it will in turn inhibit the TCA cycle and energy production. We have an interesting compound, fluoroacetate. When fluoroacetate enters into the cell, it is converted to fluorocitrate, and so it mimics citrate, and it binds to aconitase, and hence it inhibits conversion of citrate to isocitrate. So this is not a competitive inhibition where if we increase the amount of citrate in the cell, aconitase will inhibit, but rather Fluorocitrate, when it binds to aconitase, it forms a permanent bond, which does not usually happen between the enzyme and the substrate, and so it completely blocks the aconitase enzyme. The only time this reaction will occur again is when the cell synthesizes new molecules of aconitase. So high levels of fluorocitrate can completely inhibit the TCA cycle. Another compound is malonate, which can inhibit the succinate dehydrogenase by acting as a competitive inhibitor of succinate. And so this will block for transfer of electrons to FAD. So these are some of the inhibitors of the TCA cycle. As we come to the end, we cannot stop emphasizing the importance of vitamins. We have seen vitamins playing important roles as coenzymes in different enzymes of the TCA cycle. For example, we've seen thiamine pyrophosphate playing a role as an important coenzyme of alpha-ketoglutarate. We have seen riboflavin in the form of FAD playing an important coenzyme for succinate dehydrogenase. We have seen NAD from niacin, important for isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, and malate dehydrogenase enzyme, where in all cases, it has served as an electron acceptor. We have seen the important role of pantothenic acid as coenzyme A in the enzymes of succinyl-CoA synthetase, and also pyruvate dehydrogenase complex that synthesized acetyl-CoA. Sorry, this is alpha-ketoglutarate dehydrogenase complex. And our anaplerotic reactions, we have seen examples of carboxylation reactions, where we've seen pyruvate carboxylase playing that, and all carboxylase enzymes require biotin as a cofactor. So you can see vitamins play a very important role in energy production in the TCA cycle. So in summary, if we think about what we've been discussing about, how much energy do we get from the complete oxidation of a molecule of glucose to carbon dioxide and water? We have seen in glycolysis, 4 ATP were produced, but 2 ATPs were used, so we had a net consumption of 2 ATP. In the presence of oxygen, if all the NADH is taken to the electron transport chain, we'll have a net of 5 ATP. So the total from glycolysis, we have this 5 plus this net 2, we can have 7 ATP. In the presence of oxygen, we've seen our pyruvate will be converted to acetyl-CoA in the pyruvate dehydrogenase reaction. And the process will yield 2 NADH, which if they go to the electron transport chain, will yield 5 ATPs. The acetyl-CoA will be taken to the TCA cycle, and we've seen one cycle of the TCA cycle will give us 10 ATPs. But remember, there are 2 acetyl-CoA, so this will be 20 ATPs. So if we add all these, the 20 plus 5 plus 7 from the different steps, we'll see that our oxidation of one molecule of glucose will yield a total of 32 ATPs. Some books will talk about 30 to 32 ATPs, but this will depend on how this NADH gets into the mitochondria, whether through the malate shuttle or the glyceraldehyde 3-phosphate shuttle. We're going to talk more about these 2 shuttle as we discuss lipid metabolism. Thank you very much, and this is our end of our lecture on TCA cycle. See you in the next one. Thank you.